Okay, we might get started. So welcome everyone to one of the Collaboration of Aphasia Trialists anniversary seminars. I'd like to thank Dirk, Lisa and the C-Star group for the opportunity to speak about some of the work of the collaboration, and I'd also like to thank everyone for joining us at this time. So for those who don't know, CATs, or the Collaboration of Aphasia Trialists, is an international network of multidisciplinary aphasia researchers across 50 countries. And some of its aims include supporting international research activities, increasing our ability to assess aphasia in different languages, supporting early and mid-career researchers, and providing resources to help support the quality and reporting of aphasia research. So it's easy to become a member of CATs if you're not already, and get involved in a range of activities. The network is structured around working groups, which have a particular focus. So there are working groups in aphasia research data sets, assessment and outcomes, effectiveness of interventions, societal impact and reintegration, cognition and aphasia, implementation science, and prognosis and markers of aphasia recovery. So this is the working group that we'll be hearing from today. So we're very fortunate to have representing this group Professor Kathy Price, who is one of the chairs of the group, and Dr. Thomas Hope. So they'll be providing an overview of the group and some of its key activities over the past year. So I'll hand it over now to Kathy. Thanks, Kathy.
Thanks very much, David. Yes, so as you've just heard, I'm one of the chairs of the Prognosis and Markers of Recovery, Aphasia Recovery Group. And the other chair is Sasha Filipovic. Excuse me. As you can see here, these are the current members that we have working in the group. And we have meetings maybe 3 times a year where we get together to discuss our ongoing work and try to think of collaborative projects. I'll go into that in more detail in a minute, but the key thing I want to emphasize on this slide is that one of our key goals is to expand our members. So a hope from this talk is that we will be attracting new members to join. And I also have a target list, and many of you are on that target list. So I will be writing to you to see if you would like to join us. So David's already explained I'm giving a joint talk with Tom Hope. I'm going to let you know about the goals of the group, and Tom is going to give you some examples of research progress to date. So the goals of our group are, as you'd expect with bringing people together, is sharing our ideas, software, data and results. We've also been talking about how we might harmonize protocols and data acquisition so that we can integrate data more easily. We're talking about different reviews of the literature that need to be done, thinking about new collaborative projects that would benefit from international collaborations. A key goal is expanding our membership so that we involve and engage with as many international groups as possible to build a consensus, and then hopefully to get some collaborative funding to allow us to work together. We've talked quite a bit about sharing data from people with aphasia across groups. So, for example, sharing language assessments that monitored recovery from aphasia, sharing lesion masks, which are anonymized, and demographics, etc. And this, if possible, would allow us to find new results from existing data, to generalize our results across data sets, and to identify rules and exceptions and understand interacting factors. For example, how do predictions for outcome vary in people who speak different languages or multiple languages, and how might that be dependent on age and many other factors for which we need large data in order to look at those multivariate issues. But the challenges, of course, are the data sharing agreements. We have been trying to share data. Well, we are sharing data that we collect at UCL from the PLORA study. But each time we do it, it takes months for UCL to set up a data sharing agreement, even though the data sharing agreements are quite similar. And then we have months of delays elsewhere. So it's not as easy to do as we had hoped. There are also data protection issues, so we have to try and anonymize the data. And then the sharing process has turned out to be incredibly time consuming and costly, with the number of people involved in it too. So it hasn't been as easy. We're sort of battling away with it and hoping that things will get easier. But meanwhile, what we're proposing to do is to share predictive software. And so different groups are developing different software. And by sharing it, we can start to generalize across findings across groups to validate in independent samples. And we can also use the predictive software for new applications, for asking new questions of data, but in using similar methodology. And the advantages of this are, the data stays in the original lab, so we don't have to set up the data sharing agreements. The software can be applied by the data owner, particularly if it's user friendly. And then it can facilitate collaborations between the software developer and the lab with the data as needed, not necessarily if the software is easy enough to use. And this avoids the problems of data sharing. So this is one of our goals, and Tom will be giving you an illustration of this later with his software. With respect to harmonizing protocols and data acquisition, we have been talking about the importance of having a shared framework that we use, trying to identify what data and software are currently available across all of our sites, what are the questions we should be asking, what sort of protocols are needed to address these questions, what sort of pre-stroke variables do we need to be measuring in the participants, what sort of control data are needed, and also considering how therapy input and outcomes can be incorporated. So currently we've had meetings to discuss, to discuss an opinion paper that might actually, again, provide a consensus across different members and collaborators and bring us all together in a common agreement that we can work towards with future grant applications, either grant applications that are coming from a single lab or grant applications that are coming from the whole group in general. We'd definitely like to have as many people involved in discussing this as possible. We've discussed doing collaborative reviews, and there's interest from different members to look at different factors. We haven't actually written a review at the moment. I mean, part of the problem is finding people that actually have the time to write them. But their suggestions have, been that we look at diet, sleep, and exercise. We look at physiological markers. We look at network characteristics, social determinants of health, anxiety, depression, genetics. And here are the people that have shown an interest in these reviews being conducted, although they don't necessarily have time to run them themselves, but they want to be involved in the review process in some way. We've also talked about new collaborative projects. such as generating pre-stroke predictors of post-stroke outcomes. So in Norway in particular, there are governmental databases where they have details of participants. So you can access these pre-stroke details of patients retrospectively. So that's a potential project that might be unique to Norway and Sweden. And then another suggestion was building computational models that can simulate lesions, and AJ Halle and Tom Hope have taken an interest in that. But we're, the more people are involved in these projects and taking them forward, the better. So with respect to expanding our membership for enhancing collaborations, the easiest way to do that is to email me. I just did update the slide, but I've clearly put the version on from before that. But I'm at UCL, and my email address has a c.j.price@UCL, J for Janet. There's another Christopher Price, and I think there's a Christine Price too. So I don't receive emails if they don't have my middle and initial in there. Anyway, I would very much like to hear from anybody who's interested in joining the group or finding out more about it. And then we've also talked about collaborative funding applications. And AJ and I have been talking about a UK research and innovation partnership that would involve bringing centres together to collect data. And Swathi Kiran has also identified the Leduc Foundation, where they have an international cross-continent networks of excellence, where the US is one, and then there's another centre not in the US leading that forward. So now we will pass on to oh, there's my email there. First, if anybody wants to write it down, then please write it down, and then I'll stop sharing my slides and hand over to Tom. Okay, so over to Tom. Does anybody have any questions before I hand over? Okay, so I'll stop sharing, Tom.
Okay.
There we go.
I will start. Okay, can you all see my slides now? I started with your one,
yes,
confusing
yes,
me enough.
yes.
I lined
Great.
it up just in case.
Thank
you. Okay, so thanks very much, Kathy. So I just talked to you about some stuff that I have been doing with a few others of the collaboration. So I've been looking at treatment responses, and the background on that, which I will not labour because doubtless you all know it, is that the evidence is not fabulous for it, but everyone knows it works and everyone wants it. And so that led me some years ago to start to wonder, well, why, or what can we do about the evidence? And one obvious confound is the very large inter-individual variability in therapy response. So I began just by saying, well, if we can predict at the individual level therapy responses, even a little, then this is evidence that the intervention works better for some people than for others, which is, of course, evidence that the intervention works for at least some people. So just to make it clear what it is I am predicting. So the hypothesis is that baseline capacities or features, things like lesion location, things like baseline behavioral data, perhaps demographic features, will drive and therefore can be used to predict at least some part of the individual variance in treatment response. And there's sort of different versions of the problem based on whether or not one is predicting the immediate post-treatment outcomes or perhaps longer-term outcomes, or both together. So it's difficult, this. Those of you, I know lots of you, or at least have come across lots of you, you will know that I have done some of this work with predicting outcomes before. But of course, these data pose unique challenges. So the complexity is common to lots of other kinds of post-stroke outcomes data. But what is very crucial here is that there are small sample sizes. So they're not necessarily small in the sense of being underpowered. They're small if what you want to do is use induction to learn a model that associates pre-treatment factors with treatment outcome and then generalize that model. Because you have complex data and small sample sizes, that means that these models overfit easily, which means that they may not generalize as well as they otherwise would. And so if there's signal there, it's quite easy to miss it. On the other hand, if one is careful, I have discovered, with estimating this out of sample performance, and if one is careful with one's models, then in the end, as we say in England, the proof is in the pudding. If it works, and assuming you're not estimating it in a horribly biased way, then it works. And my results so far suggest that it does work. So these are some earlier results. These are predicting treatment responses for treatments for alexia, that is reading impairments, and anomia, naming impairments. So each of the scatter plots represents one patient, predicted versus empirical. In both cases, if the predictions were perfect, then the dots would be along those diagonal lines. So they're not perfect, but they're definitely a lot better than chance. What you see, the brain images are the heat maps, the parts of the brain where damage was most relevant to these predictions. So I've then extended this to include a few more treatment data sets. So this is 4 using the same methodology, exactly the same as was published previously in 2021. And you can see it broadly works. Again, it's not perfect, but remember, these are very, very small samples. So until joining the collaboration of aphasia trialists, what I was limited to really was just showing that this is better than chance, just showing that we can do it at all. The joy, the genius of the collaboration of aphasia trialists is it has connected me to lots more people, including Katharina Breitenstein and Helmut Olbrich, who conducted a very large-scale trial. 108 patients is enormous, in my experience at least, for treatment studies in this area. Lots and lots of outcomes, of course, still very complicated data. It has required a little bit of adjustment of how I run the models, because these trials often have quite a lot of missing data, and you need to be quite careful in how you handle it. I think I now have quite a nice way. The key point is that in these data, because with 108 samples, you get into a zone where I can start to optimize model fit. So not just asking, can we predict this stuff at all, but can we predict it well? How well can we predict it? And the answer is pretty well. So this is a scatter plot across all 37 outcome variables, with the ANELT time points 1 and 3, that is immediately post-treatment, and then after long-term follow-up. Those are the 2 highlighted. Now, if I was sitting in the audience seeing a scatter plot like that, I would assume that someone had made a mistake, and I'm quite confident I haven't. However, it is worth pointing out that in this case, the baseline scores for these things are strongly correlated with the outcome scores. So this scatter plot is tighter than it would be if you just use the baseline scores, but it would be pretty tight looking if you just use the baseline scores. So that's why it is so beautiful looking. So again, on the right, you see a little brain map. I won't go into exactly how I calculate these, but it's not a terribly novel method. It's a relatively sort of classical methodology. The key point being that you expect most of the business areas of the brain to be blue, because more damage should equal less improvement or poorer outcomes over time. One of the nice things, as I said, about a larger sample size is you can start to optimize model fit. I have gone away from feature selection in the past because you tended to overfit in feature selection space, but in this sample I have got some quite nice results. So this is backwards feature selection. The blue line is the training data, if you like, and the red line on my screen is a test set that was not used at all in the training. So the point is that in this data set, it's slightly larger, it allows you to actually start to really optimize the model, which, it makes it a little bit better, but the key point actually might be from the perspective of science that allows us to focus our attention on a subset of the available features and therefore to better understand what it is that's driving treatment responses. So as Kathy mentioned, I think we have tried a few different ways of running these collaborations, and lots of them have been quite slow and painful, despite everybody putting in their best effort. I'd like to think I was, at least, and I'm pretty sure the people I was working with were. A lot of these data analysis things, it's very difficult to share. It's very difficult to sort of train somebody else to code up my methods. It's quite difficult for them to send me the right data. To make this faster, it seems obvious that I need to distribute a toolbox. So that is underway. And essentially what I'll do is I will knock it up until I am at least broadly happy with it. Then I will share it sort of directly with CATs members and work with them to make sure that it's not filled with bugs. And then when it's not filled with bugs, I will make it public. Conclusions so far of this work. So firstly, there is signal here. On this, I'm very confident. And the collaboration of aphasia trialists has, at least for me so far, been an ideal forum in which to do this work, because these studies are extremely resource intensive, and I need the data to work with, and I need the expertise of the people who can run these trials. I'm hoping that the public toolbox will help to speed up and standardize this kind of analysis. And I would just end by echoing Kathy's point that it's not just, I think, about applying the funky new code to people's data that we already have. What I would like to see is that the collaboration approaches what I think of as the kind of the money result, the real result that I suspect patients really want to see, which is predictions that actually help people to choose which treatment to have, which at some level must mean that we try different therapies with the same patients. So that will be, pushing for that will be my contribution to AJ's opinion paper. So that's all I have. Thank you very much. And I will just end with the advert for the next one.
Wonderful. Thank you very much, Tom and Kathy, for that great overview. And yeah, just to promote the next C-Star lecture series, and there's an email there if you want to be joined to the mailing list for these seminars, and they're also available. The recordings of each seminar are available on the YouTube channel and via the C-Star website. So the next topic on this is spatial and temporal neural dynamics for sentence production and comprehension by Dr. Laura Giglio. So we have time for a few questions before we wrap up. I might start off. So one of the, I suppose, challenges if we are wanting to look at larger data sets, clearly it would be best to have harmonized outcome measures, but are you at the moment thinking of ways to be able to combine different data sets with different outcome measures, that there might be some way, at least as an intermediate step, to look at outcomes when people have used varying outcome measures, as they often do?
I'm not sure who that question's to, me or Gavin.
Well, you go, and I'll add something if
So yes, the answer is certainly yes. There are a few little statistical tricks that you can play with ranking in scores, which I've seen used quite well, not least by some of the people who are already members of the collaboration of aphasia trialists. And similarly, if you are predicting scores for which your model is not specifically trained, then you always have the option of expressing your results not in terms of absolute error, which might be inappropriate, but in terms of correlations or variance explained of the scores by the predictions, which is something that Kathy and I and others did recently in a letter to Brain, where we tested the disconnectome symptom discoverer model. And it's not trained to predict scores that we have. It's not trained to predict comprehensive aphasia test scores, but it's trained to predict scores that are expected to be close cousins of those scores. And we found that the variance explained approach works reasonably well.
Right. Kathy, did you have anything to add on that?
No, I didn't. But as Tom said, what we can do is to see how the predictions on one set of data just work on another set of data. Because if the training's done on one set of data, you can still apply that to multiple sets of data, and then you can pool those results, across data sets. So I'm sure there are good ways of doing it, as Tom's explained.
Great. Next up, we have from the C-Star viewing party. Tom, will the toolbox you are developing combine neuroimaging and behavioral predictors in the same model?
What I'm doing is I'm trying to make it so that you can do it however you like. So I think you'll have to provide at minimum some tabular predictors, at least one, and some tabular dependent variables, at least one. And then if you also provide, so I mean, that would allow you, for example, to provide lesion load variables or disconnection variables derived in your own way from MRI. But I will also include in the toolbox a thing that will essentially calculate the ones that I use from your lesion images if you tell me where they are, basically. So the point is that the toolbox will be distributed with essentially a little lesion load calculator. Simply because I think quite a lot of people have those data. So I'm aiming for maximum flexibility. It is obviously that slightly complicates the design of the toolbox and makes it more likely I will make coding errors, but hopefully you will all tell me where they are when you find them.
Thank you, Tom. Do we have any other questions from the viewing party?
Nope, not from the viewing party. Thanks. Thank you, Tom, for the answer.
No problem, though.
And thank you, Kathy, for the talk.
Pleasure.
Any other questions from anyone online? Oh, you go, Kathy.
No, I was just going to say that, we hope to hear from some people, and I will be reaching out to people too.
Yeah, and I think this has been a really good demonstration of some of the advantages of CATs with getting international collaborations, and there's clearly a great incentive to be involved with being able to get some access to some of these wonderful toolboxes that I think will help us all in advancing this area. Kathy, do you have, in terms of the other review areas, so that was one of the other activities is around those different reviews.
Mm-hmm.
Do you see that that will then lead to some of the predictors identified in those reviews then being built into other models?
I hope so, yes. Yes, I hope so. So I think, we've, Sophie Roberts, who works with Tom and I, has been looking at education effects and found that the number of years of education is also soaking up some of the variance in the outcome measures that we use. And I think that was only possible to identify with large numbers of subjects that allowed us to validate it across different lesion sizes and groups, and to look at when education has an effect and when it doesn't have an effect. So these reviews of finding things in the literature are useful for us then writing into the opinion paper exactly what we think should be included. So I think the whole thing will come together. There are so many parts to be integrated, and we've known this for many years, but actually there's so much for every group to do in setting themselves up, that working internationally, where we can say different people look at different things and then we try and integrate the results in a constructive way, we can hopefully mastermind the problem. Because it's not just about having big data, which is extremely expensive and challenging to acquire. It's also about sort of coordinated and systematic approaches to examine all the variables that might be affecting outcomes.
Wonderful, thank you. So we might wrap it up then. We're at the end of this seminar. I'd like to thank Tom and Kathy for providing this overview of the some of the very exciting work of this working group. And just to echo Kathy's thoughts on, please encourage you to get involved if you're interested at all. It's very easy to join CATs, and then you can indicate that you'd be interested in this working group in your application. And also thank you very much to C-Star for the opportunity to be presenting with you on this topic. And thank you all for attending.
Yeah, thank you very much.
Thanks for listening.
Yeah.